Welcome to planet Earth. Earth is home to one of the world's most incredible forces, life. Here we see a beautiful scene found in nature of a lily gracefully swaying in the wind. All of the organisms pictured have processes that allow them to survive. For example, all of the organisms perform a type of cellular respiration. All of the plants pictured perform photosynthesis, including the beautiful lily swaying in the wind. We're going to focus on the actual process of photosynthesis and the steps that go into it. Let's zoom into where the photosynthesis occurs in the lily, otherwise known as the leaf. This is the leaf, the main location of a plant in which photosynthesis occurs. The leaf is made up of main skin-like membranes, collectively called the epidermis. On the underside of the leaf, there are openings called stomata that open and close to release oxygen and to take in carbon dioxide. In between these thick, waxy skins is the mesophyll layer that contains a vein, which acts as a means of transport and water supply, and mesophyll cells, which are the cells that primarily perform photosynthesis. These cells contain a large amount of chloroplasts, the organelles in which photosynthesis occurs. Let's take a closer look at the chloroplasts inside of these cells. This is the chloroplast. As you can see, it is a membrane-bound organelle with a double membrane. The stacks of green discs that are seen are the grana, with each disc known as the thylakoid. The space between the thylakoids and the grana is filled with a thick substance called the stroma. It is here where the light-independent reactions occur, and it is in the thylakoid membrane where the light-dependent reactions occur. Let's first take a closer look at the light-dependent reactions that occur in the thylakoid membrane. This is the thylakoid membrane, where the light-dependent reactions occur. The purple bell pepper-shaped contraption is called a photosystem. The green circles are chlorophyll molecules, which reflect energy from light sources. Light-dependent reactions begin with a ray of light known as a photon exciting a chlorophyll molecule, causing energy to pass through nearby chlorophyll molecules in the form of electrons until it finally reaches the reaction center and transfers into special chlorophyll A molecules waiting. Water is also broken down by an enzyme during this process, so the electrons from water breaking down are transferred into the special chlorophyll A molecules as well. These special molecules then transfer the electrons into a primary electron acceptor, which prepares the electrons and sends them on to the next photosystem. The electrons are then transferred from the first photosystem, which is called photosystem 2, into the second photosystem, photosystem 1. All light-dependent reactions start on photosystem 2 and then proceed to photosystem 1. The reason for the strange numbering is because photosystem 1 was discovered first and then photosystem 2, despite their different orders. After leaving photosystem 2, the electrons are transferred through an electron transport chain beginning with the electron carrier plastokinone, abbreviated with PQ. The electrons are then transferred to a cytochrome complex, and then finally to a protein called a plastocyanin, abbreviated with PC. The process of going down this electron transport chain produces ATP, the cell's source of energy. The electrons are then sent into the reaction center of photosystem 1 and then into the special chlorophyll molecules. These molecules are the same as the ones from photosystem 2, differing only in the fact that the position in the membrane is different, causing different absorption abilities. At the same time, a photon of light excites surrounding chlorophyll molecules until the energy reaches the reaction center and the special chlorophyll A molecules. The electrons are then sent into the primary electron acceptor, where they are sent down a second electron transport chain beginning with the protein ferredoxin, or FD. From ferredoxin, they are sent to the NADP plus reductase, where they reduce a NADP plus to become an NADPH. This NADPH is then sent to the Calvin cycle for later usage. In order to power the Calvin cycle, there must be ATP available for usage. As a result, all of the hydrogens from the surrounding environment and from the now broken down water are sent into the thylakoid space, where they are funneled through an ATP synthase to produce more ATP. This ATP is then sent to the Calvin cycle to be used. The Calvin cycle. The Calvin cycle is a process that takes place in the stroma that actually makes the glucose. The cycle shows one CO2 cycle with the individual molecules. However, the numbers next to the molecules show how many of each there must be in order to keep one G3P. First, CO2 enters the cycle and combines with a 5-carbon structure called ribulose biphosphate, otherwise known as RUBP, with the help of an enzyme called Rubisco. The gray represents carbon and the yellow represents phosphate groups. The new 6-carbon structure is very short-lived and separates into 2 3-carbon structures. Then, ATP phosphorylates these 3-carbon structures to now have 2 phosphate groups. NADPH then removes these phosphate groups to form an extremely important 3-carbon structure called G3P. The G3Ps are saved until there are 6 in total, in which 1 is kept and 5 are sent off to be used again and recycled as RUBP. This process repeats itself until there are 2 G3P left over, which are then combined to create the one and only glucose.